Hey guys, welcome to the lecture for chapter 24, the origin of species. This chapter is going to focus on exactly what it says, where species come from and where we get those. So we know that Charles Darwin went to the Galapagos Islands, and we know that he was there looking at landforms. And when he was looking at the landforms of the islands, and a lot of other places that he went on his trip on the Beagle, he determined that islands must be something that are newly formed. It's not something that necessarily breaks away from another landmass sometimes, and in that determination he had the thought that maybe that means that the species might also be new too. And if the species are new, well, because they're not found anywhere else on Earth, then how did they get there? So we're going to look in this chapter on speciation, which is the origin of a new species. That was kind of the focal point of Darwin's theories, his evolutionary theory, and what he published in the origin of species. So evolutionary theory has to be able to explain how a new species is going to originate. We've got to have some explanation of the mechanisms, not just for change, but we need an explanation of the mechanisms for the creation of a brand new species. Now, microevolution is what we have been dealing with so far. We've been dealing with Hardy-Weinberg, gene flow. These are changes to the allele frequencies in a small population or a large population, but it's still just one gene pool. Right? But macroevolution is going to look at a bigger picture. We're going to be looking at broad changes, things that are going to be above the species level. And the reason why is we need to be able to explain differences and similarities between different organisms. Now, one of the organisms that kind of gave Darwin pause on the Galapagos Islands, he discovered a lot of plants and animals that were nowhere else on Earth. But this one in particular was one that he noted in his diaries, in his journals. This was a flightless bird, so a bird that does not fly. So if the species that are on the island their ancestors were from the mainland and they flew over to the island, then how in the heck did a bird that doesn't fly get to the island? It must have been different from its ancestor, and so we have some sort of speciation event occurring here. So we need to figure out exactly what causes the different species to be different. Well, before we can kind of do that, we have to have a definition for what is a species. Now, typically biologists are going to look at, when they're grouping organisms, they look at morphology, which is the body forms. They look at physiology, which is the way that the body works on the inside. Biochemistry, which we know is just the chemistry, the chemicals, what's going on with the interactions, chemical reactions within the body, and then you have DNA sequences. So these are all different things that we can look at when we try and group organisms, but we still need a good definition for a species. So we say that the biological species concept states that a species is a group of populations that are able to breed. It's about reproduction. They can interbreed, and when they do interbreed, not only can they, but they produce viable and fertile offspring. Okay, so that's kind of the key there, is that, and that's why it's in red. They've got to be able to interbreed, but the offspring that they produce also have to be fertile. Okay, they have to be able to make fertile offspring. And so this was defined by Ernest Mayr in 1942, and we actually still use this definition of a species. So let's look at some different species here. I have these two birds. They're both meadowlarks. This is an eastern meadowlark, and this is a western meadowlark. So even though they look very similar, they're considered to be different species because they have different behaviors. And so their different behaviors, their different songs, are their mating rituals, and those are what prevent them from interbreeding. Okay, another example, humans. We are all very, very different. There's a lot of diversity between us, and even though it may be unlikely for people to breed from continent to continent, it is possible, and you could have offspring between an Asian woman and an African man. It's a possibility. So that's kind of what defines all of the humans as humans under Homo sapiens. That's our species. Now, in 1993, this guy at Berkeley, his name was Scott Edwards, he sequenced DNA because he kind of came across, he was looking at that question about humans because humans are very spread out. And so if we're so spread out, how is it that the populations are still considered to be one species? Because the species is about interbreeding, but there's not a ton of interbreeding that goes on between Africa and Asia. So how are the populations still the same species around the world. And so he looked at actually a lot of different organisms, and he was sequencing the DNA, and then he made these things called gene trees, which is what these are, and they were looking at the different alleles, and then where, like which population those alleles occur in, and then made a gene tree to kind of show the gene flow. And what he concluded from that was that there are a lot of species that have gene flow for a very large distance. It's just a low level of gene flow, but that still helps to maintain that as one species. So because our definition of species is based upon breeding and whether or not you can breed and produce fertile offspring, then we've got to talk about interbreeding and the barriers that there are to interbreeding, because that's going to be the key to defining our species and what a species is. So reproductive isolation is the existence of any factor, any sort of barrier that's going to prevent two different species from producing those fertile offspring. Now, we all know that there are hybrids, and you are probably very familiar with them. Hybrids are the offspring of a cross between two different species. And so an example that we all probably know about, mules. We probably all know about mules. They're the offspring of horses and donkeys, but the more exciting ones are the ligers and the tigons. There's actually a lot of controversy over the breeding of these animals, and you can check out, there's lots of different organizations, and I posted this on Edmodo. You can check out these links, but this is, I think this is a liger. But there are hybrids, and hybrids do come about sometimes, but it's a cross between two different species, but whether or not they're fertile, we'll get into that in just a second. But reproductive isolation is classified whether it happens before or after fertilization. So we have several different types of barriers that can prevent this crossbreeding and this production of hybrids. Okay? We have pre-zygotic barriers, which are going to be barriers that prevent fertilization before the formation of the zygote. So it's blocking it. Remember, the pre means before, zygotic referring to zygote. So using our word parts, it's before fertilization or before the zygote. Okay. So these are things that actually stop fertilization from happening, and there's a lot of different ways that that can happen. We can impede the different species from attempting to mate. We can stop the different species from the completion of mating, so they could try and have it not work, or they can hinder fertilization itself if the mating is successful. So they can mate, but then there's no product made. Okay? Now, these pre-zygotic barriers will maintain reproductive isolation, and some of the major ones are temporal, habitat, behavioral, mechanical, and gamete isolation. So let's kind of look at each of these. There's also post-zygotic barriers, and we'll get to those in just a second, but those are the ones that are going to occur after fertilization does happen. So if two species are able to create offspring, there are some barriers afterwards that still prevent it from forming a new species. Okay, so we're going to start over here. This is a chart that is, or a good diagram that's found on page 490 in your text, and it's got a lot of examples, and your reading guide asks you to kind of copy it down, but this is a really good place to study, a good slide to maybe print really big for you, but this is a good, I think, summary of everything that's going on. Okay, so let's kind of look at each of these in turn. So a habitat isolation, which would be when you have two species but they don't ever see each other, and that could be because they have different habitats, but they may or may not be isolated by a physical barrier. It could be something like a mountain range or an ocean that is preventing them. Sometimes the barrier is even between the treetops, the animals that live on the treetops like in the jungle, and the ones that live on the ground. So it doesn't necessarily have to be something huge like a mountain range or an ocean. It just has to be something geographical or about their habitat that isolates them from others, and so that's going to prevent reproduction. So, for instance, we have here, this is a garter snake that lives in the water, and this is a garter snake that lives on the land. So they can still be in the same area, but one living in water, one living on land prevents them from breeding together. Okay. Some other ones are some temporal isolation. That's when the species are going to breed at different times. It could be during different times of the day. It could be during different seasons. It could just be during different years. For example, the North American skunks, one species in North America breeds in late winter and one breeds in late summer. So the fact that they don't breed at the same time is going to prevent them from breeding together. Okay, you also have behavioral isolation, and those are things like courtship rituals, the way that they mate, the things that they do to seek out a mate, the things that they do to make the mate, or make the other organism mate with them. There are things like that that can also prevent fertilization or mating from occurring. Now, I would draw, if I were you on your notes, draw a line between behavioral and mechanical. Okay, because this is the point, everything from habitat, temporal, behavioral, everything so far, those are all things that have prevented mating from happening before there is even an attempt to mate. So there wouldn't be any sort of attempt yet. Mechanical isolation is when there is an attempt, but there are certain morphological, remember morphological is talking about the morph, the form, so talking about body form, so there's some sort of difference in their body that doesn't physically allow it to work. It just doesn't work because they're not physically compatible. Okay, so at this point, now we're talking about they're trying to mate, but they're not physically capable. It could also be gametic isolation. We're talking about the gamete, and that's when the sperm of one species is not able to fertilize eggs of another, so they are able to mate, everything else kind of lines up, they physically can do it, but then there's some sort of biochemical something that doesn't allow the sperm to live. Okay, so it could be pH, it could be enzymes, there's all kinds of things that could make it so that the sperm are not able to survive long enough to reach the egg and fertilize it. Okay, so these are all pre-zygotic barriers. Now, we also have post-zygotic barriers. Oh, let's do the pictures here. So these are the blue-footed boobies, which are always fun to look at, but they have courtship rituals, mating rituals that enable mate recognition, and so the things that they do signal to others in their species, hey, we're of the same species, come mate with me. Okay, so that's what we mean by behavioral isolation. And then the physical isolation, or the mechanical isolation, shells on snails can spiral in a certain direction, and so if they spiral in the opposite direction, then they're not able to mate. They just physically can't work. The genitals don't combine properly. Okay, so it just, even if they try, it's not going to work out. Okay, so now a post-zygotic reproductive barrier. So post-zygotic would be after the zygote is formed. So that means that everything lined up, their habitat, their mating season, there's no issues with the rituals and no mechanical issues. They were physically able to do it, no biochemical issues. The gametes were able to join, and we made a zygote. So now what? There are some barriers in place that are going to prevent that hybrid zygote from becoming a viable fertile adult. And there's several different things that could do that. Sometimes the hybrids are just very weak. It could be that the offspring are very frail, and so that prevents a new species from forming because they don't continue the line, basically. They would be the ones that would die off. Now, sometimes the hybrid is infertile, okay, or it's sterile. Those would be like our mules, and so that prevents new gene flow because they're not going to be able to mate at all. So that just ends the line right there. And then hybrid breakdown refers to, this usually happens with plants, and there's an example in your book about rice, but it basically means that the hybrid species works at first, but as it continues to the next generation and the next generation, it gets weaker and weaker because the recessive alleles become more and more prevalent, and so that reduces the, it either will make them weak or it will make them sterile. Okay. Now, mules, that's the example that we always remember. Okay, mules are sterile hybrid offspring between a horse and a donkey. So that's easy to remember, but that is a post-zygotic reproductive barrier. Okay, so this is all based upon kind of the biological species concept, but that's not the only definition for a species, and part of the problem is that definitions are supposed to work 100% of the time, but if a definition doesn't work 100% of the time, then it's not a good definition. Well, the biological species concept doesn't work 100% of the time, because how do we know about fossils? We don't know anything about fossils. from this definition. We don't know who or what they can reproduce with. And asexual organisms, so all prokaryotic organisms, they mate with themselves. So they don't kind of fall into this category. So we have a little bit of an issue there. So we need other ideas, other definitions. And the other ideas are going to actually emphasize the opposite concept than the biological species looks at. Biological species is based upon limitations. It's saying that they're not able to interbreed. Whereas other concepts or other definitions are going to emphasize the unity that's within the species rather than the things that separate it from other species. The morphological species concept, we've already referred to the word morphological, but remember, morphological is talking about the structural features. It's looking at the morphology, which is the structure, the body form. And so this is a pretty good one because you don't need any information about gene flow. We don't have to know anything about who can breed with who and where the genes are flowing and all of that you need for the biological species concept. So that's good. And in practice, this is typically what we use. You usually look at something and look at the form that it takes, and then you kind of place it into categories based on the species. So this is the one that we a lot of times use in practice, maybe not meaning to, but we typically go to morphology. The disadvantage, though, is that it's very subjective. So all the scientists would have to agree, and we know that scientists don't really like to agree. So this can cause an issue in trying to determine what, or to define the different species, where the lines are with the morphological differences. Another idea is the ecological species concept, and this is looking at the species' ecological niche. And a niche, if you want to write out the definition for that, a niche is referring to how the different members of the population interact with their biotic and their abiotic components. So the niche is basically what's their role in the environment. What do they do? How do they interact with the biotic and abiotic components? So it could be a lot of different things that define the niche. But remember that only one different species can occupy the same niche. So you can't have more than one species occupying the same niche. So because of that, we can define species based upon their niche because there's only one per niche. So that's great because it applies to asexual and sexual species, so that's good. It takes into account their adaptations, which is also good, because if we were to have perhaps one species or one that's living, we got one population in an area and now there's an adaptation, maybe we have disruptive selection, and so now the adaptations are leaning the evolution towards one end of my phenotypic variations and then towards the other end. And so now we have kind of two species forming as a result of that disruptive selection. And so that's great because it takes into account adaptation. Okay. The last one is the phylogenetic species concept, which is where we define a species as the smallest group of organisms or smallest group of individuals that are on a phylogenetic tree. And that's also good because we can look at sexual and asexual species so the prokaryotes aren't left out. But where do you draw the line? How do you determine the degree of difference that is required? Which I think has been a common thread with all of these different, maybe not the ecological species, but this has been a common thread through, sorry about that. It's been a common thread through the other species concepts is that you've got to draw the line somewhere, and so everybody would need to agree on something. So, end result, we stick with the biological species concept. So there are other definitions of a species, but the one we're going to move forward with is the biological species concept, which is talking about reproduction and the reproductive isolation. Okay, so we've got to look at ways that speciation or ways that separation can occur, and there are two major ways that you need to know. We have allopatric speciation, which is where there is a geographic barrier that separates a population and then the result is two different species, or sympatric speciation, which is where there is no geographic barrier. Okay, so we're going to look at each of these kind of in turn. So this is showing kind of a visual for those two things. I have here an ancestral population, one pond. It could be that maybe we go through a period of drought and now the one pond has become two ponds because the land was higher in the middle. And so now we end up with two species, two different species, and that would be allopatric speciation because they are separated geographically. But it could be that maybe there was a drought, but then it rained, and so the rain came back up, and so now we have one population, or, well, we have, I guess, it wouldn't be one population, you'd be in one lake, and you would have multiple species there for other reasons, so they're not geographically isolated. That would be sympatric speciation. Okay, let's look at allopatric. Allopatric, we call it the other country. That's from the Greek root. So allo means other, and then patra is the root there, and that means country. So allopatric meaning other country, which makes sense because then it's referring to the separation geographically. Okay, so in allopatric speciation, the gene flow is going to be interrupted. And the macroevolution, we could say that macroevolution is the effect of all of these speciation and extinction events. Now, the allopatric speciation, remember, is focused on a geographic barrier. Now, separate populations are going to then evolve independently through mutation, natural selection, genetic drift, all the things that we just talked about in Chapter 23 after the speciation event occurs. So whatever it was, whether it was migration, if it was a natural disaster, if it was something that humans did that separated them geographically, separated the population into two, so now we have subpopulations, then the rest of those evolutionary changes can start to occur, and that just makes the divide greater. That makes the species really be completely defined. Now, the barriers to reproduction that we have here are intrinsic, which simply means that they're kind of like a part of the organism, like the mating habits. Those are intrinsic things. And so the barriers to reproduction that we see are the intrinsic things, but the separation itself is not biological. Okay, that's what allopatric is referring to, that the separation between them is not a biological barrier. The reproductive isolation is intrinsic. Here's an example, allopatric speciation, and I think this is a great picture to maybe put a star next to and write this down, because the thing to remember with allopatric speciation is it really depends on the organism, because for these organisms, I have these squirrels, okay, and these squirrels are living on one side and on the other side of the Grand Canyon. Now, that's a really big geographical separation for a squirrel. Okay, they're really not that far away, but they're separated by this huge canyon in the earth, and so they can't reach each other. So they're geographically separated. But would a bird be geographically separated by something like this? It wouldn't. So you've got to remember that allopatric speciation and the geographical barriers do depend on the mode of movement for the different species. So it is different. You have to take each and every species into account. You got to look at them separately when you're looking at allopatric speciation. Now we do have some evidence for sorry about the bells. We do have some evidence for continental drift. Remember, continental drift is about that Pangea idea that all of the continents were all one landmass together, and over time they're drifting further and further apart. So 88 million years ago, we had India and Madagascar were, really, really close together, but then 65 million years ago, they've kind of drifted apart. India seems to be drifting north, and Madagascar is drifting west, and now that divide is even greater. 56 million years ago, Madagascar is all the way down here, India all the way up here, and it's moving in that direction. And so the continental drift can cause allopatric speciation. This was a study that was done on some frogs in Madagascar, and they were looking at how closely related they are. There are some species that are in Madagascar only. There are some species that are in India and in Southeast Asia, and so they're just kind of looking at the differences there. But this is an example of how allopatric speciation could occur, and I'm sure you can think of some others as well. Okay, let's look at the other type. The other type, I think, is a little bit more complicated because it's sin. The sin is the Greek root, and that means together. So that's where we get the same country name from. So this speciation happens when there's geographically an overlapping population. So they're not geographically separated, but there's still speciation going to occur. And polyploidy, maybe put a 1 next to this one, polyploidy is the first example. Okay, so polyploidy is the first way that this can happen. And that's when you have extra sets of chromosomes because there's been some sort of accident during cell division. We're talking about during meiosis, so formation of the eggs and the sperm. Okay, now, the autoploidy is when you've got one individual with more than 2 chromosome sets, but it's derived from just one species. That's talking about a failure during cell division that's not reducing chromosome number. So we know meiosis is supposed to be a reduction in chromosome number. So we got one individual that, or one species that is not reducing the chromosome number for whatever reason, mutation, some sort of error that happens, some failure of the system, results in autoploidy. An allopolyploidy is a species that has multiple sets of chromosomes, but they're derived from different species. So that's talking about hybrids that are able to reproduce. Typically, this is when you have something that reproduces asexually, because usually the issue with polyploidy, or with alloploidy in particular, when I have a hybrid, usually the reason why the hybrids are sterile is because they don't have homologous chromosomes, because they end up with a different amount of chromosomes. The chromosomes don't match up. And so they are not able to reproduce with another hybrid. But if you have a species that reproduces asexually, then the homologous chromosomes don't matter. So that's how you can end up with your allopolyploidy. And that's why it's very common in plants, because a lot of times plants can self-fertilize. And so we say that they are partially asexual. So a lot of crop plants are actually polyploids. In fact, 80% of the plant species today, they believe, were formed from polyploidy speciation, which I think is pretty interesting. So this is a diagram to kind of show you what's going on with the chromosomes, because I think this is kind of important to understand. This is when I have a failure during meiosis. So I have a 2N. Remember that N is referring to the diploid number. Okay, remember that 2N is a diploid number. So this is the failure of cell division after the chromosome duplication gives rise. And so that's giving rise to a tetraploid tissue. Okay, so now we have something that's tetraploid. Instead of diploid, it's now going to be tetraploid. Okay, then the gametes that we produce are going to be diploid, because we take this and we divide it in half when we go through meiosis. Then that is going to produce tetraploid offspring. Okay, the offspring end up being tetraploid if I have gametes that are 2 diploid ones. So now my offspring is 4N when my original parent was 2N. Okay, but that's okay sometimes because I still have homologous chromosomes. Okay, so that's kind of the key there, is whether or not we have homologous chromosomes. So this is kind of showing you the difference between the two. So I have species A, I have species B. Species B has some sort of meiotic error, and so there's an unreduced gamete that now has 4 chromosomes. It's still 2N. Then I have species A that produces a normal gamete, and it's got an N number that equals 3. So I have 3 chromosomes. And so when those two combine and form a hybrid, that hybrid has 7 chromosomes. So nothing pairs up. Nothing pairs up properly. So I have no homologous chromosomes. That's not good. Okay? But if I have this unreduced gamete with 7 chromosomes and it were to mate with a normal gamete that has 3 chromosomes, then they could produce viable offspring, because now there's a 2N number that equals 10. Okay, so if you look, they're actually matching up. The red one and the red one, red one and the red one, red one and red one, and the blue were already matched up. Okay, so now I end up with a viable offspring. So this is allopolyploidy. Okay, this is how the plants kind of work. So that can result in a brand new species, because now this species is certainly different than either of these species. Okay. The second form of sympatric speciation could also be from ecological niches. So put a number 2 next to this one. This is our second form of sympatric speciation. Could be because I have differentiation in the habitat. So I could have, for instance, a genetic change, some sort of factor that is going to allow some of the organisms in a population to exploit some sort of new resource or new habitat. So I have the North American maggot fly that typically lives on the native hawthorn trees, but now some of them, because of some, could be mutation, could be introduction of new genes, wherever it came from, but now some of them in the population are able to feed as also on the apple trees. Well, the apple trees mature faster, and so that's going to now create temporal isolation. So this is our sympatric speciation because of the ecological niche, and then you can see how next, the thing that would happen next would be the gap is widened between the two species by those other mechanisms of evolution that we've talked about. So I could have temporal isolation because now they're not going to be around at the same time because the apple trees mature faster than the hawthorn trees. Okay? The third type of sympatric speciation is sexual selection. Okay, sexual selection, put a number 3 next to this one. This one is number 3. Sexual selection is just referring to what we talked about before, the intra and the intersexual selection, and so those, typically it's mate choice that is going to drive this type of speciation. For example, there is Lake Victoria that has, I think it's like 600 species of cichlid fish, which is crazy amount of fish that live in one lake. And so why would that have happened? They do say that, or they do think that it happened because of, well, a lot of reasons. There's a lot of things going on in that lake. But they do say that sexual selection is occurring because of mate choice based on color. And so the mates are choosing, or the women, or I guess the females, are choosing their mates based upon the color, and so that kind of keeps them within their categories. The blue ones mate with the blue ones, the yellow ones mate with the yellow ones, and so that keeps the species separate. Okay, so to kind of sum up what we've done so far, allopatric and sympatric speciation are going to cause reproductive isolation. Those are the two ways. Allopatric speciation is when the geographic separation is going to restrict gene flow. Okay, then reproductive isolation occurs through the other forms of evolution, natural selection, genetic drift, sexual selection, whichever it is that's going on. Okay. Even if contact is restored between the populations, interbreeding is prevented. So that tells us that we have a brand new species. Okay, that's allopatric speciation. Sympatric speciation, a reproductive barrier isolates one subset without geographically separating them. And so that could result from something like polyploidy, natural selection, or sexual selection. Then again, the other evolutionary mechanisms that we learned from chapter 23 about natural selection, the genetic drift, the gene flow, all of those things can then start to affect the population as well. But what happens when contact is restored? We said here if there is a separation between the populations, contact could be restored. Well, what would happen if it were to be restored? What happens if there is contact between them? You end up with something called a hybrid zone. And a hybrid zone is a region where the members of a different species now mate and produce hybrids in that area. Okay, and so this is kind of a picture of that hybrid zone. This, I have the yellow-bellied toads and then the fire-bellied toads, and the hybrid zone is right in the middle. It literally is the line between where the two species live. And so it could be a single band. A lot of times the hybrid zone, or the hybrids that are in this hybrid zone typically have reduced fitness compared to the parents. Remember, we're talking about reproductive fitness. It could also be that they have developmental issues. They could have abnormalities. And so you're not going to really see a lot of gene flow between the different species because the hybrids are very weak. They're not fit. Now, the hybrid zone could be a little bit more complicated. This is a pretty straightforward hybrid zone line, okay? And the lines can be thick, they can be thin, but it's still a very distinct kind of zone between the two. But not all the world is like that. Some areas are a little bit more complicated. It could be that there are multiple habitats within the same region that the different species occur, where it's a little bit more spotty. Things like hills or rivers or things like that kind of disrupt the geography that spread species around means that you can have a multitude of hybrid zones. It's also more complex if the hybrid is actually stronger than the parents, like if the fitness of the hybrid that's formed is not weaker, then that's going to really affect the speciation that's occurring there. But the point is, the patterns with the hybrid zones tell us about the factors that influence the evolution, that influence the reproductive isolation. So it's good to look at hybrid zones because they tell you a lot about what's going on evolutionarily. Okay? Now, if I were to look at the different hybrid zones kind of over time, giving them time, you would see that there are three basic outcomes. Given enough time, one of these three things would happen. Either there is a strengthening of a reproductive barrier, we call that reinforcement, which reduces gene flow because the hybrids are less fit than the parents, and so natural selection acts upon that. The relative fitness is lower than the parents, and so the hybrids are the ones that die off because of natural selection. And so that would be reinforcement of the two different species. Fusion would be the opposite. Fusion is when there's weakening of the reproductive barrier and eventually fusing into one species because the gene flow actually reduces differences. Okay, so that can happen too, where I end up with just one species, one resulting species. Stabilizing is a little bit more complicated. Stabilizing is when you have a variety of different factors, and every single population will have different factors that do this, so it's not something that you can generalize and say, well, this is what always happens, because each and every population is different. But it's basically when there's continued formation of a hybrid individual. So you would think that it would stabilize, or that it would fuse, or that it would be reinforced, but there's something else at play that is causing the hybrids to continue to be created. Okay, so that's called stabilizing. This kind of gives you a visual of what's going on. I've got here my population. Say I have 5 individuals, small gene flow between the individuals, and then I have a barrier to the gene flow. It could be any number of things, but the population diverges, and then over time it changes. And so now the population is different. Technically, we call it a different species now, and so these are still going to be breeding within themselves. And so now we have species A and species B, and so the hybrid zone is when there's gene flow between the two different, or when there's reproduction between the two different species. And so you can end up with reinforcement, which is going to further drive the blue arrows away from each other, or it could be fusion, where the blue arrows come back together and they form one species again. Stability is when the arrows stay the same. The blue arrows kind of stay the same. Continued formation of the hybrid, the ratios and the percentages don't really change. The frequency doesn't change much. So the next question that we ask ourselves, the next logical question is, well, what's the time frame? How long does something like this have to take? Speciation takes how long? Okay. Well, we can look at broad patterns in speciation. We can look at the fossil record, we can look at morphological data, or we can look at molecular data. So several different ways to kind of look at organisms to see how long did that take. Now, the fossil record is kind of hard to look at because the fossil record includes examples of species that appear to have appeared suddenly. They persist unchanged for a really long time, and then they disappear. And so it's really hard to look at a fossil record and kind of determine what's going on. But these guys, Eldridge and Gould, Eldridge was he's a paleontologist from the American Museum of Natural History, and Gould, Stephen J. Gould, was from Harvard. And they were saying that this period of stasis, of no change, and then a big punctuation of a brief period of rapid change, he called it punctuated equilibrium, which is very self-explanatory. Punctuated, think about punctuations when I have periods or commas, that like pauses your sentence. So your sentence is going on and on and on and on and on, and you're droning on in a monotone, probably like I am, and then I hit a period, and it stops, and there's some sort of change. So it's like a change in the equilibrium. It's a punctuated change. Now that's very different from what Darwin is saying. Darwin was kind of going with the gradualism pattern, saying that there are gradual changes that eventually cause the different species to become different. So these are our two basic patterns that we see in speciation. We have punctuated equilibrium and we have gradualism. Part of the problem is that fossils are really hard to tell speciation. You can't really see what's going on with time, especially because we have well, we have issues, obviously, with fossils and time and getting the time right and looking at large periods of time. We're not looking at small periods of time. So it's kind of hard to see what's going on time-wise when you look at fossils. But also, things that separate species are not always morphological. Sometimes it's a reproductive isolation that you don't fossilize. It's not something that you can see in a fossil because it could be something like temporal, it could be behavioral, and those are not preserved in a fossil. So that makes it really hard to tell species just from fossils. So, how can we tell? Well, we can look at genomics. Genomics is a serious key to, and it's relatively new, to figuring out the different species by identifying specific genes. And you can look in some species, in some cases of speciation, you can look at exactly which gene caused the difference. In fact, some species, it's just one gene, or it may be just one allele, or it could be a combination of just a few alleles or just a few genes that cause such a difference that prevents or that creates speciation. For example, I have those snails again. Those snails and their shells, the spirals are what physically make it impossible for them to breed together. And that spiral is caused by one gene. So there's only one gene that's responsible for that. So if that changes, then boom, we have a new species. So you can use genomics, you can use DNA sequencing and looking at the different genes and at the different alleles to give you a little bit more insight into the speciation events and how long it takes for all of these things to happen and what exactly it is that's causing the speciation. But the big idea here, the big takeaway, the main idea is that macroevolution is a culmination of speciation and extinction. We're going to look at extinction in our next little chapter. We've already kind of talked about it a little bit, but we're going to look at extinction events next. So you've already got the speciation part. Hand in hand with that, speciation, the creation of a species, is extinction, the extinguishing of a species. So big idea, speciation is super important when it comes to macroevolution. Big changes, rapid, not rapid, but broad changes over long periods of time. Make sure that you know the difference between allopatric speciation and sympatric speciation, and then kind of the different ways that each of those can happen. Also, be sure that you kind of understand this idea between the ancestral species and the products here, and what can be different between them. This is that example of the polyploidy. Make sure you go back and review that, and let me know if you have any questions. These are kind of the things that I want you to be able to do. You need to be able to tell me what's going on, what are the different four species concepts, how do we define a species, why do we go with the biological species concept instead of the others, but what are some of the advantages to those, as well as the limitations. Be sure to tell me the differences between pre-zygotic and post-zygotic reproductive barriers. That diagram on page 490 is really great because it's got examples, and there's a chart in the reading guide that kind of puts it into a different perspective, but that's a really good way to look at definitions and examples. And then be able to tell me between allopatric and sympatric. That's a really big takeaway from this chapter. And then the polyploidy is also really big in reproductive isolation, especially because that happens a lot. The hybrid zone, and then the 3 outcomes for the hybrid zones over time. We will be doing a brief recap in class, so make sure that you made note of any questions or any issues that you have with this chapter, anything that needs to be clarified, so that you can let me know in class and we can discuss it, and then we'll move on with some group activities and some labs.